Zinc finger domains are structural motifs used by a large class of DNA binding proteins. They use centrally coordinated zinc atoms as crucial structural elements. The zinc atom is coordinated by 2 cysteine residues from the beta sheet and 2 histidine residues from the alpha helix. A single zinc finger domain is only large enough to bind a few bases of DNA. Zinc fingers are often found in tandem repeats as part of a larger DNA binding region. The helical region of each zinc finger rests in the major groove of the DNA. Amino acid side chains project out from the helix and contact bases in the DNA. The identities of these side chains determine the precise DNA sequence recognized by each zinc finger. Assembling different zinc finger motifs allows precise control over the sequence specificity of the protein.